All right, hello. Let's go through a roundup of the Pfizer BioNTech vaccine. This is where we are right now, and this is kind of some of the scientific diagrams behind some of the stuff that I've been saying. So if you wanted more detail, here's the more detail, and I'll try and keep this pretty quick and light. First of all, in some of the earlier things I said about this vaccine, I think I had indicated that it was only the the receptor binding domain of the spike protein. I don't know where I got that. Reading the new paper, it seems clear that it is the full SARS coronavirus spike they're using to vaccinate. And so some of my concerns about a very limited amount of different parts of the vaccine that could be shown to the immune system, those would no longer apply. And I don't know how I got the wrong end of the stick, but I definitely did. So, yeah. Let's see. The nicest thing, I think, about this graph is if you look right up here. So the red line are people who got the vaccine, and the blue line are people who got a placebo, not the vaccine. And so you can see the blue line, it just increases steadily. And so this is the people in the study gradually getting more and more and more virus. And yeah, it seems to follow a pretty straight line. And that just looks like the world in general. And yeah, that's fine. That makes sense. And then if you look here, the red line for vaccinated people starts out exactly the same. So they're getting the virus at exactly the same rate. And I think this worked out so perfectly that essentially they've shown the little blow up here because they are very proud of this little bottom bit of the graph. And yeah, justifiably, I think. So you can see that the real turning point seems to be somewhere here around 12 days, or if we're being, hedging our bets, let's say maybe 2 weeks. There's probably no difference between vaccinated and unvaccinated. And after that, yeah, the number of people that get the virus who've had the vaccine is actually very low outside of that little bit. Other interesting part is that these people who are protected in this range right here, up to 21 days, have only had one dose of the vaccine. They have not had the second dose yet. So this says the first dose is protective in most people, but the protection doesn't kick in for about 14 days. And the second dose is going to pick up anybody who maybe wasn't sufficiently stimulated in the first dose. And then potentially give all the people that did make a good response in the first dose a much higher, longer lasting immune response. And that's a pretty good thing. Yeah, so actually, step back one step. Of the people that did get the virus, most of the ones that got the virus after, I think, the 21-day period is the ones they are counting. There was about the same rate of severe COVID to symptomatic COVID. It's just that the overall incidence of disease was much lower. So you could say that, yeah, this does seem to prevent some disease, but it won't necessarily protect everybody from severe disease. It is, like they say, something like 90% effective. What's different about this vaccine? Well, past studies had shown, two past studies had shown that if you have regular messenger RNA, this will have some effect in sort of waking up certain parts of the immune system. Kind of depends on which cells it runs into, how many of that kind of cell there are at the site, we think. And the results are fairly variable. I've got a slide coming up that shows this. But the Pfizer vaccine substitutes this, which is the U nucleotide, uridine triphosphate, for this thing, which is a thing called a pseudo-uridine. And it's got the same face over here. So this is the part that would actually bind to the other nucleotide. And so that is the same. It's just that the rest of it's a little different. The idea is that this might be a little bit longer lasting, or that this may not be recognized and may not provoke an immune response. So we can look at the science behind that. So here is so the actual science behind the Pfizer BioNTech vaccine is not published for the most part. They have been very focused on just getting through the trials as fast as possible and not exploring interesting little side items and interests. But we can look back at some of the science that led up to the vaccine. So For example, the vaccine is being delivered in these things called lipid nanoparticles. What is that? Well, it looked like this. It's a little drop of essentially lipid, like fat, with a little bit of polyethylene glycol, which is something you would find in just about every toothpaste and shampoo in the world. It's kind of a generic thickener that we use, holds things together nicely. And twisted up somewhere inside of this, they have a little bit of RNA. And all you do is you mix a vat of RNA with a vat of this lipid and a little bit of the polyethylene glycol, kind of shake it up or, do something like that that's very low tech, and you end up with little blobs that, for the most part, are going to contain all of the parts, the RNA and the lipid and the PEG. So, what do these things do? Well, so this paper was looking at the difference in delivery, like how well it works. And so they're showing that, so here they have unmodified RNA with the regular U nucleotide versus pseudo-U RNA. And they're trying to express a couple different things. And so when they try it in cell culture, using the real thing seems to work a little bit better. Not horrendously better, but something like twice as well. You get about twice as much expression. But when you put these things into an actual animal, then it seems as though you get roughly the same amount of delivery if you're using the pseudo-uridine or the regular uridine. So it's pretty much a wash. Yeah, it's certainly not any worse, and if it does limit side effects, then I could see that as being a pretty good thing. So here from a nice 2016 paper, they are summing up the evidence. So there were 3 earlier papers, 2 from the same group. They only did a very limited study of what parts of the immune system were being basically annoyed by the RNA. And they were using the RNA to express different proteins. So these were expressing a beta-globin gene from, I believe, a frog, Xenopus. And these were expressing an albumin, and these were expressing human growth hormone. The earlier studies found that the modified one was a little more effective than the unmodified. The one more recent study found that the unmodified was more effective than the modified in mice, and they're looking for expression in the spleen. And this other study shows that they are just about the same, and it actually looked at a much broader panel of different ways that the immune system could be annoyed and really couldn't find any meaningful difference there. So I think we've got just about equal evidence on either side of this, and probably I would tend to go with this one. As far as I'm aware, the Moderna vaccine uses regular U nucleotides, and the Pfizer vaccine uses pseudo-U based nucleotides, and there's probably no real difference between these two. If there is a difference, it's so small it is not showing up in animal experiments. Okay, so what happens at 14 days? Yeah, there it is. The question's right at the top, because that's about the point where we seem to be protected. Unfortunately, in the previous studies of the Pfizer-BioNTech, this is the phase 2, the best data from that, I believe, they looked at 7 days and they looked at 21 days. So you know that whatever you have at 7 days is probably not enough to protect. And you know that whatever you have at 21 days may be enough to protect. And so the answer is going to be somewhere in between if you're looking for what is the amount of immune response that I need in order to achieve protection, roughly, because it's going to vary from one person to another. So here we go. The dose that they settled on is going to be the sort of brighter green one right in the center, which is a 30 microgram dose. That's what was actually approved, and they are giving it still at 1 day, or day 1 and day 21, so 3 weeks later. And they are looking. So at 7 days, these people basically had not made any demonstrable immune response. At 21 days, right before they got the vaccine, they had an immune response. So the average was about 29 neutralizing titer. And this is a particular kind of test where you try to stop a virus from getting into a cell using antibodies. You put the virus with the antibodies, and then you see how much virus can get in without any antibodies there, and how much virus can get in with antibody there. And then you dilute the antibodies down until they don't work anymore. And so you can dilute the antibodies down about 30 times, and then they no longer work. And so it's a 30-fold dilution, is I think what they're talking about here. And so, yeah, it looks like once you get to about 30, 29 in this one, you are probably protected. Are you protected at 20? We don't know. Are you protected at 15? We don't know. At some point, you're not protected. In terms of total antibodies, and again, these are just antibodies to the R receptor binding domain, the RBD. It looks like about 1,500 is a reasonable level, although they're measuring it in the very weird units per mil designation, which none of the other papers use. But you can see that's roughly comparable to what you would get from a real infection. These are people that have had the virus and got over it, although notice the little dot down here. Some people have the virus, get over it, and still do not make much of an immune response. So, there we go. All right. What are the side effects of the Pfizer vaccine? Well, the side effects are these. Let's see. The green are mild ones. So this is a temperature that is essentially not raised, and it's probably just like soreness of the arm. Then the blue ones are moderate. This is a temperature that is raised by half a degree or so, maybe 1 degree Fahrenheit, but not really enough that you could tell. The yellow ones are the ones where I would be a little more concerned. These are people that have a temperature rise of maybe 2 or 3 degrees. So these are people that actually get a fever. Now, these are not particularly high fevers, and it appears from the paper that these were fevers that could be and were controlled pretty well. You notice that the rate of fevers, you got some mild fevers even with the placebo injections. So, yeah. And I don't believe there were any grade 4 events that show up in this. If there were any, there might have been one somewhere, but I do not see it. And so grade 4 would be a more serious kind of event, like an anaphylactic shock kind of thing. So, yeah. This is based on something like it was around 25,000 in each of the treatment groups, neighborhood of 45,000 overall. So a lot of people, and yeah, that many events without a severe adverse event. I think so far there have been 6 anaphylactic events where they'd want to give you something like an EpiPen out of, it's something like the first 280,000 vaccinations. That's a really low rate, and yeah, they still do not know why those 6 people responded differently than the other 280,000, but chances are it's probably something more to do, I would say, with the people and or just a weird coincidence of which cell got hit first rather than something to do with the vaccine itself. So these look really safe as vaccines go. All right, so when you get your vaccine, where should you put it? Yeah, should you mainline that right into your veins, or should you like inject it under your skin? Should you stick it into a muscle? Where? Yeah, intraperitoneal, that's like into your abdominal cavity. So this is looking at how much of the protein was expressed. So like this is what the vaccine's trying to do, make this protein. And yeah, you did get the best expression when it went right into the vein, but the expression did not last very long. So you had the shortest half-life when it went into the vein, and you had a half-life of just barely over a day, actually a little bit under a day when it goes into a muscle. So half of the RNA is gone within the first day. You're down to less than a quarter of the RNA after the second day, down to less than an eighth after the third day. By the time you get to a week after the vaccination, there's essentially nothing left of the original inoculation inside of a person. So people that are worried about long-term effects, it's hard for there to be a long-term effect when there is literally nothing left that could be causing the long-term effect. I think the closest that you could get, and this is still really speculative, would be if a person developed some kind of immunity against They're looking at that polyethylene glycol right now, but like I said, you were exposed to that probably last night in the shower and this morning while you were brushing your teeth. So, yeah, the odds of that being strongly immunogenic and reactive, I don't know. Did your head swell up when you took that shower? And or did your teeth and gums just start to swell up when you brushed them? I'm not, not talking about gum disease here. Talking about anaphylactic shock. It seems really unlikely to me that this is going to be anything that's widespread or dangerous. So it looks really safe and doesn't last for long. And if it doesn't last for long, yeah, it's really hard for a thing that is no longer there to have effects down the road. I mean, you get what I'm saying? Yeah. So I think this is one of the nicer features of RNA vaccines, that they are made of a super destructible substance, and so they are also themselves very short acting, short lasting. There's not really any way that they could hang around for longer or do anything sort of down the line, months or years later. Other people are asking, what are the long-term effects of vaccination? And that's a good question. This is the first data we have on this particular anti-COVID RNA vaccination. And so this is from Moderna, and they looked 4 months after the first dose, which is about 3 months after the second dose of vaccine. And what they show is that, the amount of immunity seems to be pretty stable, particularly in younger people. Looks like it drops off a little more on average in older people. And so from graphs like this, we can say that, okay, it looks like we start out, so this is a neutralization titer. So it looks like we start out at about 256. And if we're assuming that 300 is roughly the level where we're probably going to be only borderline protected or maybe not protected, and you look at the drop. And so for young people, it looks as though they drop to about maybe half that level over the first 3 months. And for older people, it looks like they drop to maybe a quarter of the original amount over the course of 3 months. And that may be an overestimate. It may be a little better than that. So, one half-life every 3 months, and it looks as though we are about 4 half-lives away from that. So 256, half of that would be 128, half again would be 64, half of that would be, 32. And 32 is probably the point at which you are borderline protected or no longer protected. So neighborhood of 3 to 4 half-lives is probably what we've got. And so for younger people, it looks as though that would be about a year. For people in these two age groups, it looks like that would probably be about 6 months. And so that is the window. That's the window of vaccination. Now, the other thing that I have heard is people saying, well, we don't know much about the long-term effects of RNA vaccination in general. And that is not exactly correct. So if we come out of this one and go into this one, here we go. It's a nice paper. Here's the title. Here are the authors. And it's actually published before all this COVID stuff started. And they have a nice little timeline here, so we can blow this up a little bit. And in this timeline, you can see that there's really a long history of RNA vaccination, or RNA. It starts out as RNA delivery to cells. The first case is back in 1989. And the first actual phase 1 and 2 human trials of RNA vaccination are published in 2008, 2009, and 2011. There are additional trials published in 2015 and 2019. And so over this time, we've got a track record of, call that 12 years plus, because you publish after your trial's been running for a while, of safety data on exactly this kind of vaccine. They were made against a different protein, so they were an anti-cancer version of this, but the RNA is the same, the lipids are going to be essentially the same, so there wouldn't be any real reason from a scientific point of view to expect that the new vaccines would be different than the older vaccines. And the older vaccines have been fine. The safety records were really good for all of these. And so, that's 5 separate clinical trials that are out there and published and show you the long-term effects of RNA vaccination on human health. And so that's what I mean. When we're looking at the COVID vaccine, we are looking at a tip of an iceberg. This is most of the rest of that iceberg. And your little COVID tip is right up here. It actually starts after this paper. So it's this little bit, 2020, after the 2019. And yeah, there's really a lot of good science behind this. A lot of people that have been asking all the questions that are going around social media, but they've been not just asking them, but asking them, doing laboratory tests on them, figuring out the answers to those questions, and then publishing the answers to those questions. And something that I learned during my PhD, just because you haven't looked up and read the paper, yourself, doesn't mean that the research and the paper on it don't exist. Yeah, that was kind of drilled into me. When somebody would ask a question, there was definitely an answer. I hadn't read the answer, and yeah, that's why you read papers, so that does not happen. So for all the people out there that are worrying, I think these are some of the papers that you can point them to, and you can find all those listed in the back of this particular paper. The URL is right up there if you want to go and type all of that in. Just pause the video and put that in. It looks like it is freely available, and yeah, great. Good luck. If you have trouble accessing this and need me to download this and put it up somewhere, I can probably do that as well. So anyway, yeah, I would say have a happy holiday season, happy Christmas, or whatever you're doing. And yeah, you know what? If you got a chance to get the vaccine, that is probably the best Christmas present you can get yourself and your family and your future. And yeah, soon as my turn comes up, I'm gonna be down there with my sleeve rolled up and my arm out saying, yes, please. Because yeah, it's an RNA vaccine. It's no big deal, but getting COVID can definitely be a big deal. And yeah, this is basically the cure for COVID. It's a preventative cure that means that you are going to have one less worry for a little while. And it is the tool that potentially allows us to get in front of COVID and eventually stamp out SARS coronavirus 2 from the human population for good. And I think that is definitely a goal worth working for. And yeah, something that I think we can take a lot of hope from. So, thanks very much. This has been Ask Dr. Ben.